It's great to be here, and I'm actually going to talk a little bit about experiments, quite a lot about experiments, in fact. But now that you mentioned, I still consider myself a theoretician in some ways, most ways even that, or maybe not so much a theoretician, but a person working with models. So even when it comes to experiments, I'm not necessarily super interested in those high-impact nature and science papers, but I care about the models, and I think that's the unifying factors for us. And I'm going to try to weave in both some of the simulations that we're doing with the lab stuff, and in particular how we try to approach these things as models. So you probably, as you've all been through a bunch of different talks about various proteins, systems, and everything, what I love with these proteins is that they're closely related to all the things that goes on in our brains and the nervous system with feelings. And once a long time ago, I gave a talk on the subject of sex, drugs, and rock and roll in a monastery, and I collected all these pictures. Don't search for these words in the monastery Wi-Fi, by the way. Most things are blocked. But there's a, jokes aside, there's a fun connotation here that the second picture you have up there is that it's 4,000 years old, about alcohol, ethanol. It's one of the drugs that mankind has used and abused for millennia. And that starts to weave not just into the nervous systems and our brains, but also culture, right? For better or worse, our culture would not be what it is without ethanol. Nicotine is another key drug, and up on the right there you have benzodiazepines, which is a very large part of modern psychiatry. They all hit these channels. There are a bunch of rock and roll connotations to this too. Pete Doherty is a big fan of this type of research. And when we originally got started here, it's almost 20 years old. That was when I was a postdoc at Stanford, because I had a colleague working in anesthesiology. So this is Propofol. It's one of the most widely used anesthetics in the world. It's cheap, it's efficient, it's difficult to overdose, although Michael Jackson eventually managed. And this turns out that these receptors are the entire basis of modern anesthesiology. And that's something that you can easily forget about how important it is. We all tend to ravel about cancer research and everything, but 150 years ago, it used to be the fact that a good surgeon was a fast surgeon. And the reason we can do modern medicine and surgery is that the patient is not lying on the table and screaming. And to make even more worried, we haven't really progressed a whole lot further than the Morton Auditorium studies some 150 years ago. This was all based on trial and error. And until roughly 30 years ago, surgery, anesthesia was all trial and error. We still don't really know what is it that makes you go unconscious. The cool thing, though, is that it works. So the trial part, lately in particular, has gone much better than the error part. And then, already about 100 years ago, there was a beautiful result. So when people try different anesthetics, there is a virtually perfect correlation between how hydrophobic an anesthetic is and how efficient it is to create anesthesia. These curves are so good that if a student were to hand in this, I would guess that they had cheated, right? That you don't get this type of correlations over 5-6 orders of magnitude. And based on this, there was a fairly widespread agreement that, once we discovered cell membranes and everything, that all these drugs, if they're hydrophobic, they must somehow influence the cell membrane. They would go into the cell membranes and change the fluidity or the lateral pressures or something. And that's a great idea, and for a long time, the more we learned about these systems, the more sense it made. So lately, that we've been able to isolate these actions to the so-called synapse, which is the small space between two nerve cells. So once you get to the end of one nerve cell, the signal, which is electrical inside the cell, would cause some vesicles to open. These vesicles will release neurotransmitters, which is typically just small molecules like amino acids. And they would then diffuse over this small space, and then they would bind to these blue dots here, which are other ion channels. They're ligand-gated. There are ligands binding them. And depending on what ligand's acting there, we're either increasing or reducing the flux, and that creates a nerve impulse in the next signal. And if we can then change how these ion channels work by influencing the membrane, that makes a lot of sense. Now, the cool thing with this is that there are two different ways we can control these channels. Occasionally, we can increase the conductivity of the channels by adding these neurotransmitters, and for other types of channels and other types of neurotransmitters, we rather reduce the effect. The fascinating thing is that they don't bind in the same place as the neurotransmitter. And this creates two ways of regulating your nerve system. You can imagine with a car, you could either use the gas pedal to increase the speed, or kind of always running the engine at full speed and using the clutch to regulate the speed. The latter is very expensive, but it creates an ability for you to, and it would also wear out the engine, right? And it's kind of the same thing in the nervous system. It creates a very fast response. Because if you're about to burn your finger on a stub, it's really important that you react in a tenth of a second. All this is used in anesthesia. And what we've learned the last few years is that you have a ligand binding outside the membrane, and then there is some sort of protein earthquake-like effect that the entire protein changes conformations here, and some 50, 60 angstroms away, you actually have the ligand, the pore that would open or close. And then we now know that there are a bunch of other binding sites, allosteric binding sites, that actually go inside the membrane itself. And that's where you have most of these compounds, such as ethanol or something, acting and allosterically modulating the channel. No matter how much ethanol or anesthetic you add, they will never open or close the channel themselves. And this is somewhere where we got started. That at the time there weren't even any good structures available. There were some super low resolution Cryo-EM structures that I won't even show you because they were pretty much, they had the right shape, but that was pretty much it. And then over the years we've gradually learned a little bit more, in particular from heroic groups doing electrophysiology. This all changed some roughly 10 years ago when the first prokaryotic structures were determined. And this is a strange concept in itself because prokaryotes are known for lots of things, but not their well-developed nervous systems. So it's not even clear why we have these proteins in eukaryotes. But they're fairly good models, actually. They share 25-30% sequence to the eukaryotic structures. They tend to be gated by other ways, obviously. They can be pH-gated or ligand-gated, and we don't really know what they're doing. But they're fantastic models. And they're so fantastic, so when we got access to these first structures at roughly 3 angstrom resolution, you could pretty much instantly create an alignment and then build models of the eukaryotic proteins. And that's really where I got started in my group. We'd done models before, but this enabled us to run large-scale molecular simulation and actually try to simulate what happens in gating and where do things bind. This alignment is so good that I would usually say I would eat my left shoe if it's wrong. There's just one minor gap there before this red TM2 helix. And depending on how the alignment is done, that gap can occur either before or after that TM2 helix. There is only one small problem. That alignment would rotate this TM2 helix by pretty much 180 degrees. And that's likely the most important helix in the entire protein. Still, when we did these models, if we forget about that alignment and just trust the computer for a second, it turns out that we identify a beautiful binding site just between the 2 subunits. It's a pentamer, so you have 5 identical subunits in eukaryotes, and just between these 2 subunits we can bind either ethanols or anesthetics. And not only can we bind them, we have a few key residues, serine 267 in particular, that is known that if you mutate away this residue, at least it appears that the channels won't really bind anesthetics. So at this point we kind of felt that we got this. We've been able to use simulations to model exactly where the ligand binding sites are. And I had a very skilled French postdoc at the time who ran a bunch of simulations. He was even able to show how anesthetics and alcohol enter either through the pore or the bilayer, bind specifically to these binding sites. They have residence times up to a few microseconds, and then they can leave again. And a beautiful equilibrium, in particular if it goes through the membrane. We published this, of course, and I think we made some not necessarily grand claims, but we felt we understand this in principle. And we were even more impressed when you see that you actually get hydrogen bonding specifically to this serine 267 residue, and this is ethanol in the picture there. There were only a few problems here. One of them, the good thing is that there's plenty of experimental evidence that this binding site has to be located between the 2 subunits. The bad news is that there is also plenty of experimental evidence that it's located inside each subunit. And that's the problem with experiments. There is too much of it. So that the second you have an hypothesis, you can virtually always find some experimental data that will support your hypothesis. And that is a danger that I think, at least I, as a theoretician, frequently fall into, right? That you only look up the evidence that supports your hypothesis, and then you're happy with that, and then I believe I'm correct. And somewhere along the road here, we gradually became more and more interested in the experimental side too. Nevertheless, we believe these results, and it makes a lot of sense, right? This particular is a protein, it's a model of the glycine receptor that's important in alcoholism, for instance. It makes a lot of sense that we would open it this way. you're literally pushing a wedge into between the two subunits. The only problem is that a few months after I presented this at a conference in Paris even, our experimental colleagues at the Pasteur Institute, they published a beautiful first co-crystal of the bacterial protein. Remember, our simulations were of the eukaryotic model. And they have a fantastic electron density of propofol bound intra-subunit, that is inside each subunit. And remember, 2 minutes ago, I just said that I would have eaten my left shoe if we were wrong, and I had predicted binding up there instead. And look, I've been wrong 1,000 times. I don't mind being wrong. But what worries me is that when simulations start being wrong under conditions where I was goddamn certain that this has to be right, because that means that I have no control over what we're doing, right? Being wrong because my error bars are gigantic, that's fine. And at somewhere here, I felt like dragging a paper bag or a brown paper bag over my head, stop doing simulations and move over to experiments. Thank God we didn't do that entirely, but in hindsight, there are a bunch of problems with our studies here, and that I think that we frequently do as theoreticians, that you have one setup in the experiments. They were prokaryotic, they were X-ray structures, while in the simulations we had homology models. We were trying to do this on eukaryotic proteins. It's a different protein. It's allosterically modulated in different ways. And there are simply too many unknown unknowns. On the other hand, it's goddamn near impossible to get these large experimental groups to start working in a prokaryotic model system just because I care about that. They need their high-impact papers to survive. So at the time, with some cheering by my experimental colleagues, we actually decided to set up the first laboratory part of my group, where we were particularly doing electrophysiology. And this sounds advanced, it's not really. The second you have RNA, you use frog eggs, and you don't have to perform surgery on the frogs yourself. You just order them from a company in Germany, actually. And then we inject some RNA in these frog eggs, and then you put them in an incubator or wine cooler. Wine coolers cost 1/10, and they're just as good. And then we let them grow this protein for 2-3 days. After that, 99% of the protein on the surface of the cell is going to be my membrane protein. And then you, very physically, you just push into 2 glass electrodes that you see here and measure the current flowing through the entire cellular membrane. And because 99% of it is my protein, I'm effectively only going to measure the conductance of my protein. And that enabled us to do a bunch of studies where systematically mutating the channels and seeing how is the conductance depending on the mutants we do, and in particular, how can they be allosterically modulated. And one of the cool things that we were able to show is that these channels can actually be either potentiated or inhibited. That is, we can either amplify the role with a very short alcohol, in this case methanol or ethanol, but once the alcohols get up to 3 carbons, you actually start inhibiting them instead. So that it's not just a matter of that you're changing the behavior, right, but you're literally changing the sign of the allosteric effect depending on the size of the molecule. We are able to control this and shift this towards larger molecules by doing certain mutations in the channel, but I won't have time to go into the details there. But the point is that that's really the only thing we needed experimentally. And once you do that, we can go back into the simulations and start to model this in a fairly primitive manner, actually, and map out, based on these different mutants, it turns out that you will have different pockets of different pocket sizes in different locations in your simulations. So in the wild type in particular, you would have this red pocket that you see up there, and then you pretty much don't have any pocket between the subunits. But what happens when we introduce certain key mutations that make the bacterial channels look more like the eukaryotic, you see that we open up an entirely second large binding pocket. So depending on what molecule this is, we either have a pocket inside each subunit, or we have pockets both inside and between the subunits. So in hindsight, it actually turns out I think our simulations were quite correct. It's that we had no idea about all these differences we're doing. Now, that is a horrible problem from another point of view, because it doesn't help to be right if you don't know you're right. It's easy to be right, and once somebody else has actually determined the structure experimentally, then we can confirm that we were indeed right. But we were so hesitant that we didn't even believe our own results until we had been able to confirm them experimentally. And at least for me, that is still a challenge with the theoretical way of working. And the good thing that since then, a few years later, there are now a bunch of experimental structures that have actually confirmed this, that the first experimental studies predicting binding between the subunits for the allosteric modulators were in fact right. And at this is something we've continued with. Because if we have both inhibition and potentiation, these are channels where there should basically be an on button and an off button, which is super interesting in anesthesia, because what everyone would really love is pairs of drugs that you could reverse anesthesia rapidly, so to control how deeply you sedate the patients. So we worked quite a lot of this, both electrophysiology, but increasingly with X-ray crystallography. That is the one thing I don't do in my lab. So here we work together with the Mark Delaruche group at the Pasteur Institute to really map out all these different binding sites. So for a long time, none of us could find a single protein that we would have it bind, that we would have a structure either under open or closed conditions with the allosteric modulators bound. But after several years of work, we were actually able to do that. Remember that first picture I showed you when we had binding inside each subunit? I wouldn't say that it's wrong, but it is kind of wrong, because that is not the inhibitory state. So after another 5 years of work, we were finally able to show that you can actually bind the small anesthetics in the pore. It's not strictly a pore blocking mechanism. There's rather a selected fit that once the pore is closed in a closed state, these anesthetics just happen to have exactly the right size to bind and form hydrogen bonds between two sites, and it selects the conformation when it is blocked. And the way you can see that is you get if I change the side of the side chains here, there are going to be other molecules that fit better. Now, I'm not saying this to somehow be, the point is not that the original experiments were wrong. They just pointed to some other structure. The structure was there, but it was a structure obtained under different conditions. And that's the other take-home message there, that we frequently get experimental studies that point in a strange way. And then as theoreticians, we immediately assume that we're wrong and we try to reproduce the experiments, even if they're incorrect. And here it turned out that the experimental proposed structure for blocking the channel has actually changed from being inside each subunit to being inside the pore in just 5 years. The other cool thing that we saw here, that here too we see this, not just changes in how strongly we potentiate the channel, but we see changes in signs. So we start mutating this binding site inside the channel that we originally thought was the inhibitory binding site. It turns out that if we replace this methionine down here to a tryptophan, normally that means that we can actually potentiate the channel. That is, that if we're adding an anesthetic, the more anesthetic we add, the more strongly the channel will react to, in this case, a pH jump, but it could also be a ligand present. And that works great. You go from 1 micromolar to 10 micromolar to 30 micromolars, and then something happens. When you move to 100 micromolars, suddenly the effect drops a little bit. And once I'm at 300 micromolars, the effect has reversed sign. I think we're all aware of strange binding behavior, right? But it's very... For instance, if you take a sleeping pill, what you're usually not used to the fact that if you double the dose, suddenly it would cause you to wake up instead. And that's effectively what happens here. Or do we know of any such things? In a way, we do. So it's Christmas, and you might end up having a bit of alcohol. And what happens with alcohol, in particular if you're a student or something? The first glass of alcohol, or the second, that probably makes you more attentive, right? And it makes you relax a bit. After the 10th glass of alcohol, you become tired. And so many of these drugs tend to have similar bimodal effects. So there are lots of narcotics that makes you hyperattentive, but at very high doses, they make you fall asleep. So this starts to correspond very closely to the type of behavioral patterns you see from lots of recreational drugs. And in the interest of time, I'm not going to go through all the structures here, but what we are increasingly believing is that, on the one hand, we have this conformational selection that the ligands can stabilize either an open or closed state if the protein is already present in that state. But what we don't yet understand why we get these very strange bimodal binding effects. But what if there are two binding sites? One binding site that is really, say, located between the subunits, that is normally that would open the channel. If that one has the highest affinity, you would start to bind there, right? But eventually you will have filled up that binding site. And as long as there are binding sites available, you will get a stronger and stronger and stronger effect. But once you've filled all these binding sites, if you keep increasing the concentration, there is no site remaining for these anesthetics to bind except in the pore. And then you start blocking the channel. So we've seen a number of examples of this, and what we can also do now is that purely by serendipity, Stephanie noticed that if you introduce this mutant, the methionine 205W again, we end up distorting, and this is simulation, a combination of electrophysiology and simulation results. In the simulations, we see that we distort the entire helices. So right next to this, there's a proline. So there's a proline and a tryptophan. And just for fun, we even tried to introduce another proline. So we have two prolines right next to each other in an alpha helix. And normally we would assume that this would completely screw up the protein. It doesn't. In fact, it makes it work better. So we somehow create a very deep binding site with a very strong effect. And what's even cooler is that, let's see in the interest of time, you can pretty much take almost anything here that distorts the helix, tryptophan, glycine, or proline. And then you get a very strong potentiating effect from the allosteric modulators. And this effect is not only strong. Stephanie came back a week and said, there must be something wrong with the experimental setup. Because what she can do, normally you apply the anesthetic, and then you wash it out, and then you check afterwards and make sure that things are back to normal, right? They don't go back to normal. They've done it for every single other protein we've always checked, but not this mutant. So there is some sort of memory effect, or that the protein binding site has been able to bind these anesthetics so hard that they don't really release from the protein anymore. So they stay bound for almost 30 minutes. And you can even apply the anesthetic, and then we wait with applying the pH jump for 3 to 10 minutes. And after 10 minutes, the effect is still there. So what... sorry, and the reason why this is even more interesting is this is an effect you see in anesthesia. Many of these, you have very long half times when you induce anesthesia. It can take a minute or two for the molecules to bind. Now the problem is that once they are bound, it can take a minute or two for you to reverse this effect if we realize that we've sedated the patient too deep, which is a significant problem. We have been able to determine quite a lot of structural and functional work of this, but the neat thing is that we believe that we finally have some pretty neat models for how this works. So what likely happens is that we have effectively a reservoir in the membrane for the anesthetics. And that's why it's so important to have this Meyer-Overton correlation. Unless they are hydrophobic, they will never enter the lipidic reservoir. Once they are there, they will bind predominantly, well, they can certainly go in through the pore, but they can also bind from the side into these binding sites. And what we effectively did here is that we opened up these binding sites to the membrane, made them more accessible. Once they go in, well, they're already hydrophobic, so once these molecules go in and bind there, they will tend to stay bound in this binding site. And even if we wash them out and everything, there will still be a reasonable concentration of the hydrophobic compounds left in the membrane. And that's likely that you have a remaining equilibrium in the membrane of these things that can last for minutes, even though I washed everything away up in the solvent outside the protein. In the interest of time, I'm going to continue a little bit more. So this far, I've mostly talked about the transmembrane part, but there is a lot of fascinating things going on extracellular. So this is work together with Chris Oelen's group in the Netherlands, in particular Mieke Nuis and Özge from my team. So this is another small prokaryotic receptor, ELIC, but it's cool because here we see another bunch of different binding sites up in the extracellular domain, chloropromazine or bromopromazine. And depending on where these ligands bind, they tend to, you effectively have an a beta sheet. We might see it better there. We have a beta sheet that is normally structurally disordered. And once these ligands bind between each subunit of these beta sheets, the beta sheets effectively closes up. So it becomes a very well-packed binding site, but in particular it means that the beta sheet is now making interactions with the next subunit. So we're effectively creating a small link from one subunit to the next subunit based on the simulation results. And what these links will then do, if I go back to this slide, they will cause the entire extracellular domain to twist a bit. And this is what we've seen in quite a few examples of structures between the difference between the open and closed state. What likely happens is that you have this twisting and blooming motion of the extracellular domain, which pushes on the transmembrane domain, and when you push on the transmembrane domain this way, this causes all the helices to twist a bit, and that is eventually what opens or closes the pore. So this is really the first examples, I think, where we can start to, we're not seeing the entire conformational earthquake in a simulation, but we're seeing the early stages of is how the transmembrane domain is starting to twist. We've continued with a number of studies like this. So just a few months ago, we were finally able to run the first simulations and structures of really beautiful eukaryotic systems, the nicotinic acetylcholine receptor. So these are human receptors, well, they're mice, but they're effectively human. They're much more complicated. They have large extracellular domains. They have large domains outside the extracellular domain, and we also have the first example of the intracellular domain. What makes these systems more complicated is also they normally don't work in your body unless they're assembled of different monomers. So that there are some famous examples when you sedate mice that for GABA in particular, the interface between the beta and the alpha subunits, that's where they have the anesthetic binding site. And if you mutate that away, you can't sedate mice. So when it comes to these systems, we were able to use simulations to, again, explain what actually happens in the Cryo-EM experiments here. We were able to identify both the water binding site, the ion binding sites, and the nicotine binding sites, and also show how ligands are actually, sorry, not ligands, how lipids are interacting with these systems, in particular cholesterol. And the cool thing here too, we're seeing these effects as ligands bind. So here you see nicotine bound. In the blue subunits, we do not have any ligand bound. And you see how they're really floppy. And in the green subunits where you have these ligands bound, you again see the beta sheets closing on the ligand. So we see exactly the same activation mechanisms here now published with Cryo-EM 2 years after our original simulations, which is quite sexy. I really love this when we can use experiments to confirm our simulations. We're frequently wrong, but now and then we're right. In the interest of time, I might skip a few more results on this so that we actually saw some beautiful pathways of the ions here, but I'll have to wait for that. But to me, there's, as much as I love these things, there is still something that I'm missing because as I mentioned before, I'm fundamentally a modeler. I like to understand concepts. And as nice as the structures are, predicting specific things doesn't really help me to understand why these are moving and how they're moving and how we can use computers to predict that. So Laura Oriana in my group started doing something very simple that normally we only start simulations for one structure, but what happened then over the last 10 years here, we got more and more and more structures. And you can kind of use the experimental space of structures to just draw up a map. What are the states that these structures would like to represent? So if you have a whole ensemble of structures, for instance of a prokaryotic protein, then you can start just plotting what are the principal components, not in any simulation, but purely what is the experimental data? What degrees of freedoms are present in the experimental data? Now, once you have these degrees of freedom, you can start very simple, pretty much Brownian dynamics, so that if we now force the protein to, if we drive a simulation to go from one of these open or closed states and follow these low dimensional 3, 4, 5 degrees of freedom, how would they move and what states would they visit? And in this particular case, it turns out that we can map out the prokaryotic channels here, what are their degrees of freedom, and all the different experimental structures, where would they be placed in these low dimensional landscape. If you now start to perform simulations to go between the open and closed state, it turns out that they tend to visit not all, but quite a few of the important so-called locally closed intermediates. So as simple as these models are, and these are beautiful simulations to me because you run them in 20 minutes on a web server, at somewhere we appear to be able to sample some sort of reasonable structural transition path. It's definitely not exact, but it might not be completely horrible, and at least we're probably not having any horrible clashes. Of course, there is no way the energetics here is going to be correct, but now we have some sort of tentative pathways that we could try to refine, and that allows us to use much more powerful methods and simulations in particular. So what Catherine Berg and my team has been doing is that she's been using Markov state models now with these channels on massive scales. We've been running hundreds of microseconds to 10 microsecond long simulations. So literally take these initial paths and each of the dots there, either in blue or orange, seed Markov states models with that, and not just one simulation per dot, but 10 different simulations. So Markov states models is effectively a way to start by grabbing single shots, such as if it was from movie images from a movie, and then we try to cluster this to describe what structure or what intermediate cluster are you really sampling in each of these, and then we effectively try to sort these into populations. So what are all the important clusters being sampled, not just in one simulation now, but in our hundreds of simulations. And after that, we start to identify what are the transitions between these states. And if we just sample this well enough, I don't need any individual simulation to sample all the way between open and closed. I just need to be able to sample transition between adjacent clusters. And if that is possible, we should be able to develop a model for how all this is moving. This is very preliminary data. Full disclosure, we don't have enough sampling yet, but this is starting to look quite impressive. So I just felt I had to share this with you. This is now again a low-dimensional space, just the 2 lowest degrees of freedom. But in the Markov state models, of course, this is a full-blown molecular dynamics simulation. I'm just using this for the clustering and to show you what it means. So let's see, up on the right, that's the closed state at pH 7. And then we have a trough, a low free energy minimum there that at roughly -1, -1 along these 2 first coordinates. That corresponds quite closely to the closed state, the closed experimental state. And that's great. So we're predicting that to be most stable. If I move to pH 4.6, that's where the channel is supposed to be open. That looks bad. So it has certainly changed a bit. There is a small population down there up to the right where you might have a local minima that actually corresponds to the open state in the simulation. But the simulation still predicts that the lowest free energy pot is still the closed state. So the simulation is wrong. Or is it? Because this is where the theory, we don't know whether it's wrong. Because we like to think about an open state channel, but it's not the case that if the neurotransmitters bind to your cells, it's not that every single channel in every cell of you will be open 100% of the time. If they were, you would die because you would completely exhaust the ion imbalance in your proteins, right? So that in the body, what happens is that these channels, they open transiently, but then they close again. So what we are increasingly believing based on experimental models that at any point in time, it might only be, even under so-called open conditions, it's only like 5% of your channels that are actually open. And suddenly that corresponds quite closely to what we see up there. A small population that's open, but the vast majority of them is still closed. But to really test whether we can predict things, we decided to do a mutant. So we took an isoleucine and replaced that with a threonine to make the channel slightly more hydrophilic. And this particular mutation, we know that it will shift the channel so that it will open at higher pHs, so it should be more open. And the cool thing is that we see that. So now already at pH 7, we see a small population in an open state, and at pH 4.6, we've also expanded the open state. So not only, both we and others have certainly been able to simulate gating before, right? But here we can use simulations to predict the effect of the allosteric modulation. Not just the binding of the ligand or the pH shift, but the secondary small effect of binding an anesthetic. Second, I can actually show what happens when I, on top of that, start to do mutations in the sequence. So that suddenly simulations are able to at least get the qualitative result right about the secondary effects now, mutations or allosteric modulation, which is something that, at least in my hands, we've never been able to do before. And just to show you how cool we think this is, up at pH 7, this is a magnification of that side. Do you see 2 small blue dots there, right in the trough? Those are the experimental closed X-ray structures. So without starting from them, the Markov states model specifically predict that the closed state structure do correspond to the lowest free energy minimum in the simulations, which is kind of remarkable. I think that this was pure luck. Had they been within 10%, I would have been really happy. So for all their shortcomings and everything, I'm not going to say that I always trust simulations. There are many cases where the simulations are right, but they're wrong. But I'm also astonished in many cases how good simulations are. For all the issues we've had with the experiments here, in many cases I would say that the more experiments we started to do, the more impressed I've been with simulations. To do this completely right, one challenge here is that depending on the pH states and everything, we end up with slightly different Markov models. To do it really properly, you would have to use the different Markov states models based on the different pHs and structures and then project the other ones on them, but I think we're getting into too much detail there. The curse for us here, though, which is something that we haven't solved yet, is the sampling. I think Catherine has something like 500 microseconds of simulation data here. There's a lot of simulations on GPUs. And we still don't, we're getting, I think, at the top right there, we have a reasonable convergence. But this has to do with how many degrees of freedom are we picking. We can pick lots of degrees of freedom, typically distances between pairs of atoms and helices, but we have too many degrees of freedom. So then we need the computers to sort this out and try to figure out what are the most important degrees of freedoms in your systems. And you can use this with variational autoencoders and everything. There's quite a lot of fancy machine learning techniques to do this. And I think we're getting there, that on the top right, we're starting to see convergence, but the other three sets, when we have slightly fewer degrees of freedom there, we still don't have convergence. So these are very powerful methods. They will allow you to sample dynamics up to multi-microsecond scale, but it doesn't necessarily mean that you can do it over lunch. Now, the good thing, as much as I swear over this in the team, I try to console myself with the fact that in a few years there's going to be so much computer time available that we won't know what to do with it, and this is what we're going to do with it. So I think the computer problem is one that will be solved, not trivially, but we will have orders of magnitude more power there. When I was a PhD student, the longest simulation in my entire thesis was 100 nanoseconds for 8,000 atoms. The second last part I'm going to tell you about is Cryo-EM. And this might sound like a complete shift, which it isn't. The reason we got involved in Cryo-EM, actually, it was a complete shift initially. The reason we got involved in Cryo-EM is that we set up a facility for this in my department in Stockholm. And then we scratched our head because my experimental colleague who had drafted the first budget of this, they figured we need some computers. So they had reserved 250,000 euros for that. And then when we were over in Cambridge, they just laughed and said that you're off by at least one order of magnitude. So we had no idea how to handle the computational problem. And now we're not talking about simulations, just taking this image data and reconstructing three-dimensional densities. Since we worked in GPU acceleration for a very long time, this ended up as a side project in my group that we worked together with Jorge Jerez in Cambridge to rewrite all the stuff that we could do it on GPUs. Which was a side project. I was just firm that we're never going to start doing Cryo-EM. I'm not very good at saying no, because suddenly I had this really good student in one of my classes. We just talked about the importance of undergraduate teaching. And I gave roughly this presentation to them, and then she came by I want to try to do a Cryo-EM structure, one of those channels. And she was really good. This was just going to be a thesis project. After, no PhD or anything, but she can spend 5 months trying to purify that protein. Yeah, after 6 months, she had this. So this is grids. And you see the small rings here? Those are channels. You see the channels from the top and the small hole there is the pore. I am in shock and awe about this. I know how you probably know how Cryo-EM works too, but the fact that we can actually see individual molecules in a microscope. I can't understand it. I understand it that it works, but that it actually works in practice. And then I was not very good at saying no, because then she also got funding to do a PhD. She got external funding for that. So, of course, by now we are doing experimental Cryo-EM on these channels too. I wish I could say that this was due to me, but it's entirely due to Urska. So we now have sub-4 angstrom structures of this channel, definitely in the closed state, which we haven't published yet. It's going to be submitted after Christmas. But we also have structures at different pHs. We're now at the point where we can use Cryo-EM to really try to go after the entire ensemble. What happens when these channels are gradually opening and closing? And one of the hottest questions for me now that we're trying to answer with Markov states models, but where I don't know the answer yet, is that do they actually move symmetrically or not? In all structures, they will always be symmetric, but that's because the structures are averages, right? We all know in computers, things are more stochastic. So I would almost bet that the way these motions actually have to happen in practice is that you have one subunit at a time move in and out. But this far, we haven't been able to show it, likely because our ways of classifying the structural data is not good enough. And that's where these two things start to come together. Yes, I will have time to finish this, just a few more minutes. In principle, what you do in Cryo-EM and simulations is very similar. And this is what I love with Cryo-EM, that in Cryo-EM, just a simulation, you have an ensemble of things. It's noisy, and there's a question how you're performing these averages. There are completely separate questions what happens when you freeze things and everything. That there's a long story about how you perform the punch freezing and how you do the actual data collection that I will leave aside. But there is a corresponding parallel on the right-hand side there. How do we treat the averaging and everything in the simulation? There are shortcomings in the models, but Ideally, I would like to compare the ensemble of how molecules are moving in the test tube that I might be able to probe with the Cryo-EM with the ensemble of how things are moving in the Cryo-EM data. The way we do this now is through a bunch of maths that I won't have time to go through in detail, but in principle, this is all based on probability, that I want to calculate what is the probability of observing a certain structure based on the whole experiment. And there's a bunch of math there involved, but basically, based on the images I observe, we assume that nothing happened in plunge freezing and the image statistics is all nice and we don't end up distorting anything. Fat chance. There are a bunch of methodological problems here, but it probably kind of works. The only problem is that this is a computationally intractable problem. So you would need to go through every single possible image, and for every single possible image, and there can be a million of them, you would need to test every single possible conformation of your molecule. Forget about exascale computers, you would need zeta or yotta scale computers for this. So to handle this in practice, the only way, if you would like a Nobel Prize, you should solve that equation. What we have to do in practice is that you have to introduce three-dimensional densities as an intermediate latent variable. So we first take the experimental data, group that into 3D densities, which is not even a 3D density, it's actually a Coulomb potential of the electrons rather. And then based on that, we then try to fit our structures into these 3D densities. And all this is done with, in particular deriving the 3D densities is done with Bayesian statistics. And for a long time, both we and others have tried to find ways. What we do now, once we have the 3D densities, we use a very advanced method to determine how the atoms are placed. And this method is called PhD, which has a beautiful analogy with Heidelberg. Once upon a time, many years before those of you who were younger, there was one of the best secondary structure predictions that was called PhD, Profile Network of Heidelberg. But I once had a colleague who actually asked that he assumed that it meant that you let the PhD student do it. And this is exactly what we do here. So you have a poor student sitting for a month and placing atoms in a map. And there is no reproducibility, there is nothing. And what we, of course, would like is for a computer to do this. So what we have developed the last 2 years, Christian Blau in my team in particular, he's just stolen the entire approach we use in Cryo-EM and formulate this as Bayesian statistics instead. Given a certain map, which is the molecule, or the ensemble of molecules most likely to have generated this map. And in the interest of time, I'm going to skip a few equations there, but we basically model the entire scattering problem. What happens, and from that, we derive a potential. So what this equation effectively means is that you reduce this to kind of, I think it's a kind of beautiful combination of experiments and simulations, that the simulation, the force field here is the prior. If I don't know anything else, I believe the physics. But I do know something else. I have observables and I see some structures. And then given those observables, how should I adjust the prior to see what should the structures really look like? And then I find a way to combine the experiments and simulation data, but in the very same model. And this works quite beautifully. It has much nicer convergence properties compared to just targeting the electron density or something. And if you just run a small simulations, these simulations finish over lunch. It takes an hour. And then I can go from, say, 7 or 8 angstrom away from the density, and the entire structure just snaps into the electron density. So instead of having a PhD student spend 1 month on it, we can do this 10 times a day now. So we've done this for all our proteins. So now we're using molecular mechanics fitting into these densities, which are beautiful. It's a little bit embarrassing because they're even better than some of the students, at least in fitting. My very best students, they beat this, in particular Urska. But the simulations here, they fix all the things that are clashes and everything, while the program makes sure that we actually do get beautiful fits. And it even allows us to do things like spectral fitting, that depending on the resolution I have in the experiment, I can apply a lot of weight with the low resolution data, the shape of the molecule, while for the very highest resolution things, I trust the simulation more when it comes to, say, placing hydrogen bonds. So I'm almost out of time, so I'm going to have 2-3 slides where I show you the final thing we're working on. The problem with Cryo-EM is that we're still freezing things. So we're at 100 Kelvin. And what we really would like to do is having techniques where I can study in particular the pores of these channels, their shapes, but ideally at room temperature. There are very few techniques that can do this, but there is one new thing that in theory will enable it, and that is neutron scattering. So with neutron scattering, you don't have any resolution. There's no way we could afford neutron diffraction and get structures of these channels. We simply don't have enough data. But I already have structures. What I just want to know is what is the rough shape of the channels, or ideally the size of the channel. And you can do this with small angle neutron scattering. This is bad in a way because it's very low dimensional experimental data. You just get a single dimensional curve roughly describing the shape. But because I already have a structural model, I can again ask this question, which model is most likely to have generated this data, and then see, does my hypothesis agree with the experiment or not? And what Marie has from this fall, we can actually say that the resting state model does indeed give the best fit to the experimental data at pH 7 of all these different models I have, both of the prokaryotic and eukaryotic structures. So you see that we fit the blue curve by far the best, in particular in this intermediate region that corresponds to the rough resolution of the structural shift we're actually seeing. And then I wish that I could say that we get, and that I conclusively say that the green curve here, that the open one fits better at pH 4.6. Well, not quite. It's certainly still compatible with it, and I can say that this yellow super open eukaryotic structures, they do not correspond well to the prokaryotic states, which is good because that's not what we'd expect. But at pH 4.6, I can't really distinguish whether the open or closed structure fits it best. So we're still working on that. But one of the reasons for this is that they're building this gigantic new European spallation source in the south of Sweden. So give this 4 or 5 more years, we're going to have 10 times better flux with the neutrons, and that is going to take down these standard errors. So in summary, that as I mentioned, I'm a model person, and I like these proteins because they're simple. They're certainly super important physiologically, but they're simple in the sense that they undergo relatively simple gating motions. But as simple as they are earthquakes over 60 angstrom. And all the things I talked to you about here was that you have resting conformations and open conformations, and I think we're gradually getting to the point where we understand these transitions and that we can predict them with computers. The thing that I haven't told you about and that we're increasingly working in the future is the desensitized state, because that's what happens when these channels open transiently over a few seconds and then they close. For some reason, they're not being inhibited for a minute or so before they can kind of recharge. And we have no idea what those states look like. And that's what we're going to go after in the future. So with that, I thank you very much for your attention, and I'd be happy to take any questions for 9 minutes at least. Thank you.